Every year, 300,000 babies with sickle cell are born in Africa, and prostate cancer affects over 74,000 men in sub-Saharan Africa annually. But genomics can change that. I am Dr. Lauritta Chinazaekpere Ndufeiya-Kumasi. I have a PhD in medical biochemistry and immunochemistry from the Department of Biochemistry, University of Port Harcourt. And I'm celebrating 2 years of PAPERI with you. So, I'll be addressing the question, how do we integrate genomics into African healthcare? We make it practical. And by that, I mean we test what we can treat. See, we don't need whole genome sequencing in every clinic. What we need are low-cost panels for sickle cell detection and epigenetic biomarkers that can detect cancers early. In Africa, most babies with sickle cell disease die before age 5 because we miss the diagnosis. But tools like the hemotype scan are changing that record. It's a simple deep-seq test that tests hemoglobin X, C, and A in just 10 minutes. And why does it work for Africa? It's affordable, it's stable at room temperature, and needs no lab, no fridge, no complex equipment. We can run this in maternity wards and immunization clinics, and we detect sickle cell at birth. We can start prophylaxis and drosy early, and that alone cuts mortality rate by over 70%. This is genomics in primary care, saving lives today. Now, let's talk about prostate cancer. It is the leading male cancer in sub-Saharan Africa, with over 74,000 cases every year. And most men are diagnosed too late for cure. Genomics gives us epigenetic biomarker. For instance, the METI scan that detects DNA methylation early stage. So here, the chemical changes that alter gene expression without changing DNA sequence itself are being screened. As we know that keeping in mind that cancer cells carry these patterns long before symptoms appear. So if we validate METI scan for African population, we can screen men using blood or urine samples. It is less invasive and it can run in regional hubs. Early detection means curative treatment, not late-stage palliative care. So how do we make this real across Africa? We do that in 3 ways. 1, we deploy point-of-care panels like the hemotype scan that I've mentioned earlier in primary healthcare clinic and maternal child health programs. 2, we build regional hubs for tests like the METI scan and connect them to district hospitals through sample transport and telegenomics. The third and the last method is by training clinicians and counselors to act on the results, because a test without a treatment plan doesn't save lives. This is the hub and spoke model. It builds expertise across the continent without duplicating expensive equipment in every hospital. Africa doesn't need to wait for perfect system. With affordable tools, smart integration, and investment in people, genomics can reach patients now. So from newborn sickle cell screening to early cancer detection, this is how we make precision medicine real for the continent. Thank you and happy 2nd anniversary to PAPERI.